Okay, I'm going to give you some information about the microbiological monitoring of natural and synthetic samples of water that we are the microbiological topic that in the way that we are working at the university to accomplish the project. In that. Al respect of that, we are working in two different topics. One is the evaluation of the disinfection level of treatment systems, and the other is the characterization of the natural water. For the evaluation of the treatment system prototypes, we are in this moment working with 4 prototypes, different prototypes. One is the prototypes, the model of prototype marks 2 that we are seeing before in the lab is this. This is the mark. The other is the model, the Udemy model, and Sawyer model, and finally the Mesita Azul that is coming with us after the visit that we were doing in Mexico. As you can see here, all the prototypes have different basal. Some of them work with different amount of water, have different time retention, if you see in all the models, have different average flow, and in general, all of them are evaluated using E. coli in one special concentration, and we are using the one specific turbidity and humic acid for all the prototypes. Then for us, the microbiological team that is working accomplishment the project, we are 2 objectives principal in the work. First is to synthetic water preparation, and the other is the evaluation of the disinfection level. In the first topic that to prepare the water, we have the challenge to prepare different amount of water depending on the prototype that we are evaluating. With that amount of microorganisms, specifically the E. coli, this strain, that is the strain that we have in the lab. And for the evaluation of the disinfection level of the systems, we try to accompany the evaluation of the effectiveness of the prototypes, that to try to take the decision for the rest of the equipment of the team. For the water synthetic preparation, we are using McFarland. It's a very common strategy to prepare the water. For that, I think that all of us know about it. We start with the E. coli, and final, we do all the validation of the McFarland technique, obtaining finally a McFarland curve, and with that, and using the spectrophotometric absorbance evaluation, we can prepare the water, the amount of water that the team needs. It's not so difficult, it's only a formula, and it's something that is easy to do. And for the microbial quantification, in general, we are working with the treatment system and biodiagnosis of the natural water. Using specifically for all of that, the membrane filtration process, that is the technique that is more common in this kind of process. For that, we do serial dilution of the sample. That depends of the system treatment, but because we need to know how much effectivity we are getting with the different systems. For that, we do the dilution, and we go with them, we do the filtration, and after that, we have the dishes and trying to count the coliform forming units between 30 and 300. This is finally the work that we are doing with the systems. Here are the conditions of this, the Mark II. In this case, we are evaluating 4 different filter strategies, combining different kind of filters, smooth and plated, with different size of poro, and using in some of the combination activated charcoal. And for all of that system, we are evaluating the quality of the water in different points. The first valve is evaluate the water before to come inside of the system on the treatment. The third valve evaluate the water after the filtration. The system of filtration, that system with different combination. And we use one final valve that evaluates the water after the UV treatment. And in this case, we are evaluating the lamps that have 12 watts. In this case, I'm going to show you some of the results of the different evaluation that we are doing in the lab with the different combination of the filters. And if you can see here, after valve 1, this is the concentration, the initial concentration of E. coli that we are putting inside of the systems. And this is the concentration of of E. coli after the filtration. If you see here, the reduction is almost nothing. But when you use the lamp, you can see the reduction is completely. Then is the lamp who is getting the disinfection of the water in the Mark II. For the prototype UDEM, this prototype is a prototype that has a dynamic sand filter. We are evaluating the inside of the water in that way. The amount of E. coli, this is the amount of E. coli that we are putting inside of the system. This is the amount of E. coli that we are obtaining after the dynamic filtration. And at the end, if you see, this is the amount of E. coli that we can find after the UV lamp treatment. If you can see, the reduction is, in this case, as the first system, get it by the UV lamp. The filtration can treat the water in respect of the bacteria E. coli. This is a Sawyer filter, a filter that was evaluated at the university that used a special filter that ultrafiltration. And in this case, the system alone gets a very good remotion of the E. coli. And this is Mesita Azul, the system that we knew in Mexico, and we was evaluating them in the university with a very good result. If you see here, we put a good amount of microorganisms getting the remotion of the microorganisms with a very low reduction of the microorganisms inside of the systems. Then I'm going to show you some evaluation that we were doing with natural water using the Mark II. If you see, this is not in the communities. It's a system that is a water that is near the university, but we want to provide the quality of the water after the treatment of this natural water. That are the results as the Mark first with different filters. And if you see, it's the same answer. We get the remotion of the microorganisms not after the filters, But yes, after the UV treatment. Yeah? Finally is the UV treatment who is doing the disinfection of the water. The microbiological team of the university is a accompaniment team, the university team, even to evaluating the quality of the water of the communities. In the Universidad, this is our department, Antioquia, located in Colombia. And in that place, we are evaluating the quality of the water of three communities. That the community that we are working with them, the Curity, La Linda, and El Carmelo. And in that place, we are evaluating the water of different places, the water uptake, the water storage tanks that the people, the community use, and the water of the house. In all cases, if you see, this is a resume of something that we showed in Mexico. But only to remember you that the quality of the water is all red. It's like to say that all the superficial water that was evaluated in the community are contaminated with E. coli and need treatment. Then, finally, we are going to show you some work that we are doing at the University of Medellin in company with another university that work with genomic, the Centro Nacional de Secuenciación Genómica. In this case, we are evaluating the microbiology. We are doing a microbiological diagnosis and characterization of bacterial microbiota using deep sequencing technique, NGCS, of surface water, natural water for human consumption. This is the thing that I'm going to show you today is a preliminary report. It's not the total because we are waiting to evaluate not only if I'm going to show you the bacterial, this work is will be related with the bacterial and fungal. Then we are working in the same community that we are working inside of the SafeWater project. We was working with the Carmelo, Curity, La Linda, and we take sample of many different places inside of the communities, trying to evaluate the diversity and the quality using genomic techniques. After take the water, we try to evaluate the microbiological and physicochemical characterization and do a metataxonomic analysis. First, I'm going to show you the characterization, the physicochemical and microbiological characterization of that water that we was evaluating. Is only to show you that you can see here coliform and E. coli, and they are the physicochemical characteristic. That is Carmelo, the same for Curity. You can see a big amount of coliform and E. coli. And for La Linda, it's something similar, okay? But for the DNA evaluation to metataxonomic analysis, we do the filtration of 10 liters of water of each point on sample. Then we did the extraction of DNA. They used the kit of the NASA, soil, Kiajin for the analysis, and do the quantification of DNA extracted by the picogreen methods. After that, we obtained this for all the different samples that we obtained, only to see that the DNA obtained was sufficient for the evaluation. Then the second step was the DNA quality evaluation. For that was used the 16S gene amplification control. In this case, we use these oligonucleotides, and that led us to obtain fragments of 1,500 pair of bases of ribosomal DNA. After that, the third step was to do the metataxonomic experimentation using a deep sequencing of that ribosomal gene, the 16S. That let us, we was using a millisec platform of Illumina, generating a segment of 100 pair of bases to establishment the bacterial community, a deep sequencing experiment of the DNA ribosomal gene was performed using those regions, the region UV3 and UV4, that are a special region of DNA that have many studies and let us do the study. The second was to use the platform motor for the analysis. And finally, to obtain the bioinformatic analysis, that is the more interesting. That led us to evaluate the phylum, the class, the order, the family, and the genus of microorganisms of bacteria that live inside of the water that we was collecting. In that respect of that, it's interesting to see that in the way that you are writing to more specific groups, family, phylum, class, order, family, and genus, the information is lost. It's like to say that many of the groups that are not known. Only we can speak about some of the genus that live inside of the water. For the phylum to result, we want to result, then it's a preliminary report because we are waiting to do more evaluation of all of that. But only to show you the protobacteria, that is the more abundant phylum inside of the water. This is the group where are Escherichia coli, Salmonella, and Vibrio, Helicobacter. All the protobacteria are the microorganisms, heterotrophs that eat many organic matter. And I wanted to result here this group because it's a group composed by many microorganisms, endosymbiotic or intracellular obligated pathogens. For that is one group important to report. Then we wanted to concentrate us a little inside of the gender distribution, the more specific distribution into the gender. Here, if you see, some of group are interesting, but we want to result the Aeromonas because this group is a group very, very important inside on the health. They cause many disease, respiratory disease, and if you see in some of the the sample of the community, specifically in Curity and La Linda, are really occupying a very important group inside of the abundance. Yeah, for that is something that we want to ask us. We want to ask for all the groups of microorganisms. We result some of them because the more important on some of them that is related with the health. You can see here Mycobacterium, Chlamydiales, Clostridium are many of them in different amount, in different samples. But here, for example, Mycobacterium is present in almost all the samples. Here, Neisseria, I don't know, I was really worried about it, this into the water. I don't want to come inside of that water. And Aeromonas, you see here, in some of the samples, the abundance is really important. Escherichia coli, if you see here, is not the more important group. Then we need to ask us for many of all the rest of the biodiversity of the microorganisms that have something important in health. Legionella are many inside of the water. Then is a preliminary report of our work in the water, all with the community inside of the project. Thank you. And this is the team. I know many of you know the team. Thank you very much.
Any questions? No, I think so. Oh, yes, of course.
It's only quick. I think this last slide is really interesting. Thank you. All of the different organisms. Really interesting.
See, it's crazy.
Yeah.
I think it's almost crazy.
Because normally, when we have water treatment, we use, like I think Bill described, indicator. Okay, so E. coli is considered one.
E. coli.
One, yeah, it's considered one of the indicators.
Many.
But when we look at the project and we look at the World Health Organization treatment aims, they talk about these viruses. You know, MS2 is resistant to UV, which is really good for the project, because if we just tried to kill E. coli, it might not be effective. But if we aim to kill MS2, because it's more difficult, it takes more UV, maybe some of these other pathogens will also be killed.
We was talking about that specifically around the aeromonas. If possible to see if differential and selective culture media to evaluate some of that inside of the project, because in Colombia, if you see, it's for all place, and are place where you can get many of them.
But it shows the importance of the other organisms that we will test for. It shows the importance of making sure the technology can remove the virus.
But I don't know. I think that we don't know exactly. I know that E. coli is a model for the bacterial presence into the water. I don't know if that microorganisms have a similar comportment, behavior inside of the water. But it's something that we think that we have to ask us, because if you see the sample of the water are really important. They are many another groups. Maybe to have a safe water could be not so easy to remove E. coli. Yeah.
Oh, hi. Sorry. No, it would just make sense as well if we maybe compared the populations between Mexico and Colombia once we have the FASTQ files and coliform counts and that kind of stuff, because we could do a comparison of the populations as well. In the water. Sorry, talk too fast.
Yeah, I don't know if I understand it.
No.
I think yes. I think that is something that is good to do, to compare your result. The thing that you were talking yesterday was really good for this work, because you find something similar. With many of the microbiological and bacteria team that have some risk into the water are in Mexico and are in Colombia. Maybe we must to ask us for that.
Can I make a little short question? Just, you know, for people who don't know anything about metagenomics and things, I see the ratio of the presence of bacteria of one against the other, and we see the majority, but how this correlate? Te lo pregunto en español. Mejor.
I understood.
Okay,
I
I will finish the English and then. Sorry.
Okay.
How this correlate to colony forming units? To have an idea.
To have an idea, for that is good. If you remember, I showed you here the amount of E. coli. This is the quantification of the E. coli of the samples. It's really, it's really small. If you see that, you say, oh, you don't have to remove many E. coli. See, but if you evaluate this inside of the here, E. coli, E. coli, the other two. E. coli represent a very little amount respect of the rest of the group. Then it's not the more important group inside of the water. In terms of abundance.
Yeah, so it's expected
Yeah.
to be the same in terms of culturable bacteria.
Yeah. Okay, I'm finished.
Okay, thank you.